Here we will discuss about this MMP9 overexpression and N-Cadherin cleavage. In the previous video we have understood the relationship between this MMP9 and N-Cadherin, that MMP9 overexpression will cause cleavage of N-Cadherin. So here they wanted to see if they overexpress this MMP9, will it cause dose dependent increase in the cleavage of N-Cadherin or not. So they did this experiment on two different cell lines, NPC039 and NPC076, and they treat the cells with this PMA that is a tumor promoter, and that will cause overexpression of this MMP9, and for different time intervals. That is, when they did it for 4 hours in the presence of PMA, there is a slight decrease in the N-Cadherin, and E-cadherin signals also seems to be goes down, but in the text they said that it does not affect E-cadherin, because maybe for other times, for other time zone or for other times intervals they do not see any changes. That's why they said it. But we can see that the MMP9 increase is very minimum compared to the control, and but the N-Cadherin decrease can be seen. And when I did it for the 8 hours, N-Cadherin decrease is more significant, and the MMP9 increase is also more significant, but there is no impact seen in the E-cadherin. And when they did it for 24 hours, we can see the N-Cadherin decrease significant compared to the 24-hour controls, and the MMP9 signals is also significantly higher. And this things is true also for the other cell line, that is NPC076. Here also we can see that there is a decrease in the E-cadherin whenever it is treated with PMA, and similarly there is an increase in the MMP9 whenever it is treated with PMA, and with increase in the time for which this PMA contact with NPC cell lines, there is an increase in this MMP9. And these things they have quantified, that is N-Cadherin signals, we can see that it goes down for both the cell lines with increase in time, and simultaneously the MMP9 signal goes up for both the cell lines with increase in time. Afterwards they wanted to see if they can use this MMP9 inhibitors, the inhibitor that is BMP, whether it will cause the decrease in the BMP signals even in the case where it is treated with tumor inhibitor or tumor promoter, that is PMA. When it is treated with only PMA, we can see the high level of MMP9 for both the cell lines. When treated with BIM along with PMA, we can see a decrease in the MMP9 signals for both the cell lines. And these things they have quantified on the right hand side of the panel B, where we can see that the signals for the MMP9 goes down when it is treated with BMP, even though PMA is present, indicating this BMP can overcome the effect of the PMA. Afterwards they wanted to check its impact of this BMP, impact of this overexpressed MMA on the cleaved N-Cadherin, that is C-terminal fragment. So when they use the secretase inhibitor that prevent the further processing of the N-Cadherin, we can see the control or background cleavage of the N-Cadherins. And these things we have also seen in the first figure when we're discussing the first figure of this paper, or in the previous video. So let's come back to the results. In the panel C, when they use the secretase inhibitor that prevent the further processing of the N-Cadherin, we can see the background signal of the C-terminal fragment of the N-Cadherin. And when it is combined with this PMA, which is the overexpression of this MMP9, we can see enhancement of this C-terminal fragment, and this enhancement is more than 2 times the background. And this thing is true for, and they did these studies for two different times, and we can see that 2 different times, or we can see the 2 different cell line. We can see in both the cases the increase in the cleaved terminal fragment of the N-Cadherin goes up by around more than 2 times, and these things they have quantified. In the bottom side of the panel C, where we can see that this CTF1 for the NPC039, it goes up for the background by like 0.5 times, and when they use the secretase inhibitor alone. But when they combine the secretase inhibitor with PMA, it goes up like around 2 times from compared to the control. Not 2 times, like 1 time compared to the control in this case. And when they used and similar things they have seen for NPC06, that when they used secretase inhibitor alone, it increased by 0.5 times, and when they used secretase inhibitor with PMA, it increased by more than 1 times. Overall, it will be like double of what they have seen in the control for both the cases. So this is how they come to the point that it's the MMP9 overexpression that caused this C-terminal cleavage of this N-Cadherin, the C-terminal fragment side or on the extracellular side. There are 2 kind of cleavage going on. One is in the intercellular part of this N-Cadherin, and one is on the extracellular part of the N-Cadherin. And this MMP9 cause the fragments on the extracellular part. That is why we have seen the results for this CTF1, which is the extracellular part of the N-Cadherin. So this is what they have shown in this figure 2 of this paper. If you need to understand more about these experiments in detail and wants to see the results, or wants to go through their descriptions represented in the paper, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.